It seems to be going. I get the signal that we are live. If you can confirm.
Yes, we are live.
We're okay?
Yes.
Thank you.
Hi Julia.
Hi Martin, schön dich zu sehen.
Ebenfalls.
I will take another minute. We know that people start walking in literally on the hour. We're making announcements and welcoming our fine participants. So, welcome everyone to yet one more presentation on we're doing the math on the 8th season of FINE, and we were getting closer to completing our 4th year straight, 130 talks so far. So we thank you all. I mean, your participation and attendance continues to motivate us and has made all this possible. So good morning, good afternoon, good evening to those attending in person, those of you joining us via YouTube. Remember, if you're watching via YouTube, you're always welcome to attend via Zoom. There is no passcode, you just show up. So always looking forward to see more people at the Zoom meeting. Before I introduce our speaker today, I want to thank Yves Davidian, who presented last week. We have a very interesting presentation and followed by what has become really a signature of the FINE series, our 1 hour questions and answers. It's a lively and lovely discussion. And Yves discussed female-male conflict in hyenas. Very interesting talk that also built on some previous presentations in FINE. As I think most of you know, remember that every talk has been recorded and you can find them in the YouTube channel. We have many colleagues who are using that for their teaching, so I invite you to consider that possibility. Next week, we're going to hear from Amiel Ilani, who I see is joining us today. Amiel is in Israel, and I think it's going to be our first talk discussing hyraxes. He'll also be talking about hyenas, but we're going to hear about sociality in hyraxes and hyenas next week, Amiel Ilani. With that, I would like to introduce our speaker today, Julia Ostner, who will be discussing aspects of the long-term research project they've had in Thailand, focusing on Assamese macaques. Julia got her first degree in psychology, then she did a diploma in biology, followed by a PhD in Germany and also at the Göttingen Primate Center. Then she got a postdoctoral fellowship that allowed her to spend some time in Stony Brook in the US, where I imagine she was interested in interacting with Carola Boris and Andres Koenig, who had been studying the same population of Assamese macaques in Thailand that Julia started working with in 2005. So this is one of those fantastic efforts. They're getting to the 20th anniversary of the project, and they've been very solid going along with all kinds of research related to sociality in Assamese macaques. Not surprisingly, for carrying on with a project like that for 20 years, Julia has a fantastic record of getting funding. She's currently a professor in the Department of Animal Ecology and Anthropology at, I'm forgetting the precise name of the institute, in part because it has this very long German name, but somewhere in Göttingen, there's a university where Julia is professor in the Department of Animal Ecology and Anthropology. I think that does it for what we need. Of course, she's been very active in every single aspect you can imagine of the discipline of primatology, contributing to her valuable time to editorial work in all the journals. She was the president of the European Society of Primatology up until recently, and so on and so forth, with, I think, close to 150 publications spanning a huge range of topics and approaches to the study of primate sociality. Today, she will be presenting on the Assamese project. And so, Julia, I would like to thank you for offering your valuable time to the fine seminar, and welcome to take it from here.
Thank you very much, Eduardo, for this super nice, very humbling introduction. Yes, I guess I start by screen sharing then. Let me try. No? Yes.
Yes.
Set the correct. Should be a title slide.
Yes, you're fine. You're ready to go.
Okay. Perfect. Yes. So I guess I jump right in. So of course I want to start by thanking you, Eduardo, for inviting me, and all the fine organizers for running what has been a fantastic series. You just said it. It's the 8th, so it's for 4 years. And for putting so much effort and work in organizing this. I try to attend as much as I can, and I always learn so much. There's always food for thought. The talk last week by Eve, really super fantastic, linking, sorry, that's me, of course, linking social and reproductive power. That reminded me, of course, of our study species, where I then, of course, started to think about, well, they don't really fit the pattern all that well, and I have to think about it more. Variation in adult sex ratio also doesn't really explain the intersexual dominance steepness in the predicted way, but maybe that's, of course, more for the discussion. So thanks for inviting me today. It's an honor, it's a pleasure. And I will talk about the long reach of seasonal reproduction in wild Assamese macaques at our long-term site. And this is obviously absolutely a joint endeavor, first and foremost with Oliver, with whom I co-direct the field site, and who, of course, has been my partner in crime for many years now, work and life. So a long-term research project, of course, is the work of many, and I cannot name them all here, but I want to highlight and give particular thanks to all past and current members of the Behavioral Ecology Department in Göttingen, and the main collaborators for the research I'm going to talk about today. So that would be Michael Heistermann, who is our go-to person for all things hormonal. He runs the Hormone Lab at the Primate Center. I should say he did run it because he retired just in March now. Wolf Daniel, who is our collaborator on the gut microbiome work that I will talk about today. And then, of course, our collaborators in Thailand, Suchinda Malavichitnond and Sutirote Mesawat, both from Chulalongkorn University. Nothing would be possible without our long-term research assistants at Pukyao. Some of them have been with us and with the team for basically since the beginning. They are truly the backbone of our project. And also Miranda Swachemakers, who is our long-term research coordinator. So what I want to talk to you about today are maternal strategies, basically. So parental strategies, of course, evolved in response to the heavy energetic demands during brood care. And in mammals in particular, females, of course, have to maintain their own metabolism, and they have to provide for the developing young through gestation and lactation, which then leads to varying patterns of resource acquisition and allocation in relation to the timing of breeding. And these strategies of energy allocation have been described to fall in a continuum from capital to income breeding. These terms were originally introduced by in a paper by Drent and Dan from 1980 for the cues that influence the laying and the clutch size decision in birds, which can be primarily influenced by the bird's current conditions, so the stored reserves, the capital, or by current conditions of the environment. So that would be the current rate of income or intake, or of course any mixture of those. So later then the idea was co-opted by Stearns and applied to life history theory, so that a capital breeder is one that uses the stored energy for reproduction, whereas the income breeder uses the energy acquired during the reproductive period rather than the stored reserves. And we have, of course, classic examples of species falling on either of these endpoints of the continuum in birds and mammals, and a particular maybe striking or very well-studied example come from pinnipeds, where the different strategies, capital, income, tend to fall on different phylogenetic lines. So we have the true seals that tend to be, the females tend to be relatively large, and mothers have very intense lactation periods where females fast until weaning. So the energy both for the female and the offspring for lactation entirely comes from the stored capital. And then on the other side, we have fur seals or sea lions that tend to be much smaller, and females alternate between the foraging trips and coming back to lactate. So females provision offspring from their current income. And then, of course, there's also evidence for mixed capital income strategies in pinnipeds. So, for example, these Weddell seals, which are generally capital breeders, supplement with incoming food during the lactation when energy reserves are just not sufficient. So this capital income framework is useful in explaining seasonal breeding strategies in a range of taxa. The application is, however, a bit more complicated in species where the reproductive cycle exceeds the period of peak food availability. So in long-lived mammals, including primates, of course, where the reproductive period from conception to weaning is usually longer than a year, can take several years, of course. And in this case, the question of reproductive timing then will be on the level of the specific reproductive phase that we are talking about, that's most costly, basically, that needs to be aligned with the favorable environmental conditions. And to illustrate this, I borrowed this figure here from Dan Brockman and Carl van Zweig, applying the capital income framework to primates. And based on the assumption that lactation is the most energy demanding period in the mammalian reproduction, this capital income model defines two distinct ways of responding to seasonality in food abundance. So we would have these A's here, up here, that would be the income breeding situation where females are selected to give birth before the annual food peak, to then use the concurrent increased or food peak for lactation to finance basically this costly phase. And in this case, females use exogenous cues, so photoperiods, for example, to start the cycle and conceive basically a gestation length prior to the optimal environmental conditions. And then we have capital breeding here, the strategy B, where females respond to increases in food abundance as a cue for conception and acquire, store energy to later finance the lactational costs. If now breeding cycles exceed 1 year, or if the environment is unpredictable, very unpredictable, it may be beneficial for females, of course, to use a mixed strategy relying on both exogenous and endogenous cues. So in this case, then females basically have the added benefit over a very strict income breeder from information on this condition threshold. So to basically not invest into something that's prone to fail in the extreme. So how is this all relevant for what we are interested in? So what we are interested in are female reproductive decisions in a seasonal environment, and how these decisions have downstream consequences in terms of maternal effects on the offspring phenotype. So what we first need to do is we need to position our study species, Assamese macaques, on the continuum of breeding strategies to understand which cues are used to start the reproductive cycle and the degree of flexibility or opportunities maybe females have or don't have in their reproduction. So how can we know where a species sits there, basically? And we can differentiate income and capital breeding based on a variety of traits, and I only list a few here. So income breeders, I explained that, should align lactation to peak resource abundance to use the incoming food for this costly period. Given the seasonal pattern, the financing of lactational costs via incoming energy, there should only be little or no fat accumulation during the gestation phase. They should use an exogenous cue to initiate the ovarian activity.
Next to myself in every single program I run, so that when one soldier falls, that's me. I'm a soldier, I think, in possible.
So they should use an exogenous cue. And interannual variation in birth rate should be relatively low, so not really depending so much on rich or lean seasons, basically, good and bad years. While capital breeders should show the contrasting patterns with a conception coinciding with the increase in food abundance. They should have a substantial fat accumulation during pregnancy to then later use that capital. They should rely on endogenous, basically a maternal condition kind of cue, and there should be much higher interannual variation in birth rate. And next, I will show you where Assamese macaques fall in terms of these metrics, and then move to the consequences of shifts in reproductive timing for offspring quality. But before I do that, of course, I want to quickly introduce the project or the study site. So Oliver and I established the Pukyao Assamese Macaque Project in 2005 in the Pukyao Wildlife Sanctuary, which is here in the northeast of Thailand. Eduardo, you mentioned that we very strongly benefited from Andreas König and Carola Boris from Stony Brook University, who had established the site for primate research in 2000. They did not study the Assamese macaques, though. Andreas and Carola, they studied the Virus language there. They are St. Patrick, and then they basically invited us to study, to use the facilities and the logistics to study Assamese macaques. So we started habituating the first group in 2006. The park is part of a 6,500 square kilometer contiguous forest complex. It's very well protected. It harbors large mammals, as you can see, and a very diverse community of predators. We are following at any time 1 to 5 multi-male, multi-female groups. The groups can have very different group sizes from 20 to 100. And this basically, this graph here shows the group history, so to say. So we started 2005 habituating. First data, systematic data came in 2006. And this group we have been following ever since. It, however, split. This group split into in two subgroups. So we are also following that group. And then there was another group split event in 2020, this time into 3 subgroups. And we keep following all these groups. This is how we are adding groups. We also started to habituate a completely new group in 2012. This also split, but we did not continue that group here just because of work power reasons. We can't follow all these groups. So we are currently following 5 groups. The home range of each group is about 4 square kilometers, and this is what you can see here. So this is from 2015 when we followed 4 groups. These are the home ranges in different colors. And you see there's very, very heavy overlap, and there are also plenty of groups around these study groups. Along with, of course, behavioral and demographic data, we collect non-invasive samples for physiology and genetics. And important for today, we do monthly fruit tree phenology on roughly 700 trees and 55 species. And so we do that in the middle of every single month. Absolutely nothing, I said that before, I want to bring it home here again, would be possible without the contribution of the project's long-term research assistants who are collecting every type of data we are using. They're very instrumental. They're ensuring continuity. Some of the assistants have been with the project almost since the beginning. We all work and live at the same camp. So they're a super integral part of everything here. Pukyo is characterized by very pronounced environmental seasonality. You see this in this climate graph with 2 distinct seasons. We have a lean, dry season here from roughly November to February, and then a rainy season from March to October. Along with this, we also have pronounced reproductive seasonality with mating from October to February and birth from March to July, with about 80% of births occurring in a 3-month window. And we have a bimodal distribution of interbirth intervals. So it's either a bit more than a year or a bit less than 2. These are data from Ines Furtbauer during her PhD. So what I talk about today, the patterns and consequences of seasonal reproduction, this talk is also a story of this long-term research site, because Ines, she was actually the first PhD at our site. She focused actually on sexual selection, and because she was the first and we didn't know anything about Assamese macaques, she also had to describe the female reproductive traits. And by doing that, she found timing effects of reproduction. Because what she found, and this is why this is her work, so this is a small data set, females that gave birth early in the birth season, so that would be in April or in May, were more likely to exhibit a short interbirth interval of a year roughly, than females that gave birth late in the season and hence closer to the next mating season. So this was our first indication of energy constraints to conceive, which is in line with capital breeding, combined with the strict seasonality. And she also showed that only 1 or 2 cycles, ovarian cycles to conception, which is more an income trait, of course, we see evidence for a mix of tactics. So now, this time we wanted to do it a bit more systematic. So now I go through these metrics. So reproductive timing. So the crucial distinction between income and capital is the timing of birth events in response to food abundance. So in our monthly phenology, I said 700 trees, but we don't score the entire forest. What we do, we score the food plants, of course. And we make sure to include all plant species that are eaten for more than 5% of the feeding time budget, at least 1 month out of each year. So this is what we're scoring. And what you see here is the variation in food availability across the year based on 175 months of phenology data. And what you should take from this is that there is a seasonal pattern, but we have a pronounced variation within the year and between years as well. So on average, across these 15 years, roughly, June and August June to August are rich. They see the highest maximum food availability. However, if you look at the variation, there can be also very low food abundance in any of these months. So August can be a bad month. The only really consistent pattern we have, the predictable pattern, is that March has consistently low food availability. So if you now overlay the births in light blue and the conceptions in dark, this would be 218 births in our data set. As I said, 80% go in 3 months. The births coincide with the onset of the best season. So the costliest reproductive phase, peak lactation, is aligned with peak food abundance. So this is the classic income pattern. A capital breeder should have the conceptions aligned with the start of the rich season. This is clearly here not the case. So, but however, there's still a lot of variation that you see. So what we think they are really doing is avoiding the predictably tough time, which is March, so that they can make use of the richer season, which may be great in some year, but at the very least, it's better than the predictably bad. So what about the next trait? We do have, oh no, what we don't have is real body mass data because we don't put them on a scale, but we still have some indications of energy acquisition and physical condition from Sonia Tutu's PhD. So here on the x-axis, you see the different female reproductive stages: early gestation, late gestation, early lactation, late lactation, and neither. And what's now relevant is, so this is just proportion time, and I only picked up here on, these are compositional models for time batches. I only look at resting here. For us, relevant is that the gestating females, so the first two, EG and LG, early and late, rest less than females of other states. And this is energy intake. They have a decreased energy intake during the gestation. So this is not indicative of any fat accumulation strategy of a capital breeder. And consistent with this less resting, less intake, we also do not find an increase in physical condition from the start of the gestation, so from conception to birth. How do we have physical condition data? For a few years, not throughout, unfortunately, we had one person score every adult for 7 different traits that we could visually score, like the number of vertebrae in the tail or the hip bone sticking out, and we had an index of physical condition. And what we see is that across the gestation period, there is no increase in physical condition. So no accumulating, so there is no pattern here, basically, no accumulating energy stores for lactation. This is, of course, not surprising, given the timing of the gestation outside of the rich season and in line with an income pattern. So what about the Q-store variant activity and conception? Ines has shown, based on hormonal data, that females show very seasonal cyclicity. They only have 1 or 2 cycles to conception, and they do not resume cycling outside the reproductive season. For example, if the infant dies early, they are not resuming. They have to wait, of course, for the next year. So these are indications for an income pattern. But of course, I already gave away our hunch of a capital condition dependent breeding. And now we are in the position to quantify this potential energetic constraint on conception. And this is an ongoing PhD work by Shivani. So we first look at that in a dichotomous way. And what we find, so this is the probability to conceive as a function of whether or not the female gave birth in the same year, yes or not. So after basically just these 6 months. And the probability to conceive is, of course, strongly predicted by whether or not a female had an infant in the same year. So in the preceding birth season, the probability to conceive is much lower if the female gave birth earlier in this year. And we can also look at that with continuous age of the last offspring. So offspring age predicts the probability to conceive. And if you want to zoom in here, this is the important bit right here, right? Only when the conception was early in the mating season, and hence the birth early in the birth season. So then consequently, the infant is already half a year or a bit older. So then when the next mating season happens, so then there is a chance for a female to go for this 1-year interbirth indicator. So this indicates that lactation indeed is very cost intensive, and the females need to be able to gain sufficient physical condition again to conceive. This, of course, is a capital breeding pattern, and we can also look at that on a more proximate level. This is a PhD from Marlies Hesen. And she found that fruit availability affects energy intake and consequently the physical condition score we're looking into. And then she looked into whether the physical condition score has anything to do with the probability of a female to conceive. And yes, a female, the better physical condition a female is in, the more likely she was at the beginning of the mating season, the more likely she was to conceive. So females are constrained in their ability to conceive by physical condition reflective of a capital pattern. And so now, given this potential endogenous threshold to conceive, maternal condition-based, it probably doesn't really come as a surprise that there is a pronounced between-year variation in birth rates. So here, these are data from 11 years, and every data point basically just tells you the proportion of females conceiving in that given year. And you see there's a huge variation from in 1 year, 35 females, 35% of females conceiving, in another 85% of females conceiving. And this is a function of food availability in the forest. So where does this leave us? It leaves us here. This is a summary slide so far. So Assamese macaques follow a mixed, or also called relaxed, income breeding strategy, with females reacting both to exogenous and endogenous condition-related cues. Maybe even more than selecting the best season for lactation, I alluded to that already, they avoid the predictably worst months for it. If then the rich season indeed turns out to be good in a given year, then they may be even able to accumulate energy to be able to conceive again in the same year, as the lactation period and the period leading up to conception basically falls together. And in this way, they sustain lactation and sometimes can even go for a short IBI. I should say they don't do that often, so they're not alternating or anything. We only have about 25% of IBIs that are 1 year, but sometimes they go, and in this way, they can maximize their reproductive output. So this flexibility in reproductive decisions or reproductive timing, of course, also has consequences, because it means that females of the same group reproducing in the same season experience vastly different environmental conditions. That also means that offspring born in the same group and year experience very different maternal conditions, which then is expected to lead to non-genetic maternal effects in the offspring in terms of different traits, survival, physiology, health, and of course also behavior. So this brings me to the consequences side of female reproductive decision for the offspring. And I first want to zoom in on the extreme for the offspring. So the situation when we have a 1 year interbirth interval, and the consequences for the offspring from the simultaneous maternal investment into gestation and lactation. So from the offspring perspective here, this would be this offspring here, if you're thinking of that, being conceived after the birth of or being conceived early after the birth of a sibling, or just half a year later, there are less resources during the gestation for that offspring while the mother is still lactating another one. So this could be a problem for the developing offspring. And then on the other side, of course, for the lactated offspring, we have the problem from an early next conception of the mother taking potentially away resources from lactating to get ready to conceive and to maintain the gestation. And we tested these effects in 2 different models. Outcome was offspring survival up to 1 year. And I should say we have very high, surprisingly high maybe, infant survival. Generally, this is what you see here. Very high infant survival, but there is an effect, although a small one, for the lactated offspring whose mother is conceiving again early during the infant's development, so during peak lactation. The maternal investment into the next offspring, into conception, into gestation, seems to take resources away from lactation for the current infant. What about the other side? We don't have an effect in terms of offspring survival or infant survival up to 1 year from having an older sibling with just a 1 year interbirth interval. So if a mother has enough resources to conceive again, the offspring has no decreases in survival compared to an infant who does not have a 1 year or older sibling. However, there may be other costs apart from survival up to 1 year due to the compromised maternal metabolic state. She still has to do these two things, gestation, lactation, simultaneously. And this is a first indication here. So a female who conceives again in the same year she gave birth in. So consequently, she is lactating and gestating at the same time compared to a just gestating, non-lactating female, has on average higher glucocorticoid levels than a female that conceives after having weaned her last offspring. So there are consequences of a short IBI, interbirth interval, in terms of decreased survival for the lactated offspring when a mother conceives again, and for the prenatally developing offspring from having a sibling that still needs to be lactated, in terms of increased prenatal stress, which we measure as glucocorticoids. So these effects of maternal metabolic state, which we measure or proxy as maternal glucocorticoid levels, is something we're really interested in, and we can better understand if we now zoom out and not only look at this small sample of 1 year birth intervals, but all offspring. And so we want to ask the question, how does the environmental condition, food availability, experienced by the mother during the pregnancy, so her basically metabolic state, GC levels, glucocorticoid levels, influence offspring phenotypes? And this is PhD work from Simone Anzà, and we were interested to understand these effects not just early in development, so for the first year or so of life, but into adulthood. So we wanted to know, are there lasting consequences of this variation in mother condition and prenatal stress adversity? So based on everything I talked about so far, you should know that the developing offspring experiences very different conditions during the gestation, depending on which year and which months this offspring is conceived in. And to illustrate this for the sample of Simone, where he used 3 cohorts of infants. So infants born in 2011, infants born in 2014, infants born in 2018. They were conceived conceived in these 3 years. So you see variation on the y-axis in food availability, and here just the dates. And so these lines are the variation in food availability. These gray, what are they, dots are the conceptions, and the triangles are the births, and these boxes should just illustrate the gestation period, the first boxes of the first infant gestation period in that sample, and the last infant's gestation period. And all you should see here is that we have a huge variation between the years as well as within the years. So being conceived early in the season can be good in 1 year, can be problematic for the next. The early part of gestation can have a better food availability than the later part of gestation, and so on. And this variation is directly linked to maternal GC output. The lower food intake, the higher the GCs, the glucocorticoids. So what happens to prenatally stressed offspring? We measure maternal glucocorticoids in 3 sensitive phases: in the early gestation, in the late gestation, and also post-lactation. Because in the early gestation, late gestation, and in the early lactation, so in the post phase. To basically differentiate the effects of timing of exposure of these 3 different sensitive phases. And then what Simo did was in one 12 months field season, we measured for all the offsprings that we had prenatal information on, so 2011, 14, 18, in a cross-sectional design aspects of their phenotype. So these offspring will be in different ages. So we have an infant cohort, basically, we have juveniles, and we have adults. Yeah? It's a bit complicated, that's why I go into detail here. And then we measure the HbA activity, so the glucocorticoid out levels of these offspring at different life stages. And in exactly same design, we also measure microbiome diversity and composition. I will get to that later, but it's the same design. So we measure, so we have for these offspring, these maternal prenatal stress levels, but we then measure offspring GCs for infants, for juveniles, for adults in a cross-sectional way. And I start with the association of these maternal GC levels with the functioning of the offspring HbA axis. And what we did, we run several general linear mixed models with age and sex as control in our 3 potential sensitive phases, so the early, the late, and the post as fixed effects. And the mean GC levels in the offspring is predicted by early prenatal and not postnatal GC levels that these offsprings experience during their prenatal development. And there was no statistical interaction with age, so these effects are stable into adulthood. And what you see here are separate models for the age classes. So we have the infant model, the juvenile model, and the adults. To just basically tell you that these effects remain. So basically this tells us the higher the prenatal stress, the higher HbA activity in the offspring, regardless of age. So this is for infants, for juveniles, and for adults. So basically this means that stressed mothers make stressed babies that grow into stressed juveniles and into stressed adults. And this fact that prenatal stress leads not only to stressed young, but is maintained into adulthood, potentially creates a vicious circle, as these stressed adult females will then, of course, transfer their increased GCs directly into their developing young prenatally, giving rise to transgenerational effects. So small adjustments in reproductive timing can create extremely different prenatal environments with lasting consequences for the offspring. And it doesn't end with an altered HbA activity, because early prenatal glucocorticoid exposure also influences the diversity and the composition of bacterial species in the gut. So again, so here, this is on gut microbiome alpha diversity. And again, we did basically similar approach. All interactions with age are not significant, suggesting these effects to be stable into adulthood. So late prenatal GCs and these postnatal early lactation GCs are not correlated with any diversity measure. And of course, we also control for current offspring glucocorticoid levels. Only, and this is what you see here, only the early prenatal glucocorticoid level are negatively related to, I use here different measures of alpha diversity. And the first one is maybe the best, the most illustrative or most interesting, because this phase phylogenetic diversity measure or index, because it takes into account the phylogenetic distance between bacterial taxa and increases if the distance is large. And this then makes it more likely that 2 taxa have different functions in the gut. So what do we take from this? Increased early pre-GCs or prenatal glucocorticoids are associated with an impoverished bacterial community, which has been interpreted as a sign of dysbiosis. So basically an imbalance of the microorganisms in the gut. So we see that as a detrimental effect. And the effects, and this is a very complicated graph, I just tell you the take-home message here. The effects of early prenatal maternal glucocorticoids. We also looked at the effects of the early prenatal maternal glucocorticoids on the relative abundance, so on the composition, on the abundance of individual bacterial taxa. And what you see here plotted are changes in the abundance of all taxa that were significantly affected by an interaction of the early prenatal glucocorticoids and offspring age, so that are changing over age, the abundance changing over age. In blue, we have positive, and on the other side, in yellow orangey, we have negative effects so a decrease. So in blue, we have an increase, in yellow, we have a decrease that exacerbated or magnified with offspring age. So they became stronger from infancy through juvenile phase, through adults. So which taxa are we actually talking about that show this differential abundance? So among those with the strongest increase, so the blue ones here, are several that cause pro-inflammatory states. This we know from humans and model organism studies, of course. And among the taxa with the strongest decrease of age, these are taxa in relative abundance. We see taxa that have anti-inflammatory functions. And importantly, the opposite is not true. There are no anti-inflammatory taxa among the blue bars here, and no pro-inflammatory ones among the yellows. And this pattern may indicate accelerated inflammation. So an increased risk of age-related diseases in response to increased early prenatal glucocorticoid levels. Of course, this is not a done deal. Basically, the next step is now to understand more about the functional aspects. And of course, the metagenomics work is on the way, but not there. Another bit of indirect evidence for an association of early prenatal glucocorticoids and the immune function comes from an basically opportunistic observation during another PhD. This is from Andreas Berghandel, who also worked on maternal effects, and he always carried his camera with him in the field because he did quantify infant growth rate via photogrammetry. And because he had the camera with him and he was looking out for his focal infants, he noticed an outbreak of what's called a pink eye infection, basically, in his focal infants. So these infants developed swollen red pussy eyes, and Andreas recorded for how long. So he took pictures and he recorded of how long he was seeing clear symptoms of these infections in his focal individuals, and linked this here to early prenatal glucocorticoid levels. And the higher the prenatal glucocorticoid levels an infant experienced, the longer he saw symptoms of these infections, which could be interpreted potentially as a compromised immune defense. So combined, these results show that early prenatal glucocorticoids lead to an increased activity of the HPA axis, an impoverished microbiome community, and enhanced relative abundance of taxa with an inflammatory function and maybe deficit in immune function. So it seems that adversities experienced early after conception have very strong organizational effects on very independent systems. So lastly, what about maternal effects on behavioral phenotypes in primates? So I have to tell you right away, we don't have the data, but what we did as a start is we searched the literature for data and found only very, very few studies on prenatal stress effects on behavior. And this is in pretty striking contrast to research on postnatal environment, postnatal adversity on later life outcome. And because of this, and because of most of these studies that we found, a few did not report effect sizes. We didn't run a proper meta-analysis. We just summarized the data as a start to then come up with predictions for the different evolutionary models of early adversity and outcomes. So here you see just a graph of the number of studies that found negative in blue, non-significant in gray, and positive in yellow effects compared to untreated controls. So I should also say these are basically all experimental studies with artificial stressors, often administered stress hormones, basically. So with a lot of caution, due to the very, very small sample size, I'm not going to go into too much detail here. We can say that across the primate studies, including humans also, prenatally challenged offspring show more distress behavior. So there was not a single study that showed less exploration or activity. This would be these, and rarely more. So more distress, less explorative behavior, and less positive, so affinitive or also play behavior than untreated individuals, controls. And this is consistent with predictions from evolutionary models of prenatal stress. These effects were often stronger after early than late exposure, which matches the findings I presented on the timing effects of prenatal stress exposure in the Assamese macaques, and is consistent with a timing effect in accelerated growth rates in our study, which I, of course, didn't go into. These studies, of course, lack ecological validity because they have not assessed offspring phenotype in the environment they evolved for. And the first field studies are underway from several studies. And for our study, Ana Lucia Bayona, I think I've seen her here today, focuses on this topic. So the effect of prenatal glucocorticoids in interaction with maternal style on infant personality and behavior. And yes, this brings me to my summary, basically. So what did we find? We find a mixed strategy, a mixed breeding strategy in females that allows flexibility in face of very, very pronounced intra and interannual environmental change. We see that the costilactation is timed, so to avoid the period of food scarcity in March that is predictable. We also find evidence for condition-dependent conception reactive to the experienced environment, which might help to spare the costs of a failed reproductive attempt. So the females basically have the added benefit of two cues, basically, they can rely on. The developing offspring experience vastly different maternal conditions, which has lasting consequences for physiology and health. And we haven't looked into that, but we will. So we are also expecting non-genetic maternal effects on behavior and life history, which is basically in line with an accelerated pace of life, which may vastly increase social complexity for gregarious species, because due to this very unpredictable combination of social and behavioral phenotypes that are realized in the population, due to this, they are all conceived during different phases and have a different maternal environment. We are expecting to see a huge diversity in social phenotypes, which increases social complexity. And with this, I'm at the end. So thanks a lot for the attention. And I guess I stop sharing now.
Thank you very much, Julia, for a very, very interesting and comprehensive exploration of this topic. So as we always do with FINE, and we've come to realize that it's a signing feature of the series, we're going to spend the next, not hour, because Julia has very kindly agreed to meet with many of us. So she has several 30-minute meetings starting at 1 p.m. So our questions and answers session will go through 12:45. That gives us 50 minutes for really asking questions to Julia. As we always do, I ask you that you just simply type a question mark in the chatting window. And if you're a student, you can add an S as an student or postdoc. We like to give priority to our younger colleagues, and we very, very much want to hear from you. We want everyone to feel comfortable asking questions in FINE. So please, we want to hear from students. We want to hear from postdocs. Just type a question mark or an S, and I will be calling people in the order that you post in the chatting, but I may skip senior colleagues or those who have asked already way too many questions over the weeks, myself included, to give priority to our young colleagues attending via Zoom. Or also, please, if you are watching us via YouTube, we know that Karsten, can we have somebody, or Lindelani, somebody can help us see if we have questions posed from YouTube?
It's not working today on YouTube. There's no chat because I saw it's too late. You chose a private video. I mean, it came up for everyone. It's everywhere for people to see, but there's no chat function today, so they cannot
so
ask questions.
I apologize to our YouTube community. Apparently, I did something wrong with the setup, and you cannot chat. Definitely, maybe that will give you encouragement to join us via Zoom. It's still open. Just click on the link, and you can join us now. So we're ready to take questions. And the first one, like always, please tell us where you're joining from and your institution, so we get to know each other a little bit more. Karsten, you're the first one.
Yes, hello, my name is Carsten Schradin. I'm doing research at the CNRS in Strasbourg, France on mammalian social evolution and mainly doing fieldwork in South Africa on small mammals. So thank you, Julia, for a fascinating talk. It's amazing to see what data you get from these wild living macaques. And I have to admit, I've hardly seen from any wild living mammal such clear correlations for fecal glucocorticoid levels. That's really impressive. Still, with this very clear correlation between early maternal glucocorticoid levels and then in the offspring at all life history stages, you interpret this mainly as an indicator that they are then permanently stressed throughout their life. Now, we know that in lactating mammals, glucocorticoid levels are always increased, and they're not only glucocorticoids do not only function to fight stress, but also just as a mean to provide energy to the organism. Do you consider the alternative hypothesis that it might be something adaptive, that maybe to some extent having higher glucocorticoid levels during life might be good if you grow up in an environment where this gives you more energy and more options?
Thanks, Carsten. Very excellent question, of course. Yes, so of course we know that we are seeing glucocorticoid as a metabolic hormone, of course. But so I have several answers, and I try to organize it now. So the quick answer is this is why we're also looking into health outcomes, which gives us maybe a clearer benefit disadvantage answer here. And this is why and all the microbiome data points into a detrimental, a disadvantage effect here. The question generally, whether this is an adaptive response or just a developmental constraint, I would say we can only answer this in the long term when we are looking at the trade-offs. So we are seeing our idea, which needs to be tested in terms of putting it all together in terms of a trade-off answer, is we're seeing an increased growth rate, for example. We are assuming that these individuals go on a different trajectory. They might speed up their life history pace. We have not yet looked into lifespan, just because we don't even want to look into it. We want to wait a bit longer, and then we will do that. So we are expecting them to go on a faster pace of life, basically have earlier menarche, start maybe earlier and die earlier, basically. And this comes at a cost. This comes at a compromised immune function, which Andreas' data at least are consistent with. The microbiome data seem to be consistent with this. And it might come at also compromised behavioral, social, cognitive skills. So I can't really answer to whether this is an adaptive response or whether it's just a developmental constraints and maladaptive. This is something we are going to look into the next years, really targeting on the trade-off level here.
Okay, thank you.
Thank you, Karsten and Julia. I'm coming next, and my question relates to Karsten's. I don't know if I missed it. Do you have any controls for the glucocorticoid levels? But I was also thinking about the microbiome, because, and I don't remember if you discussed this, would looking at females who are not getting pregnant at the same time that you're looking at the pregnant ones be one way of controlling for the case you're building? And the same with the microbiome. I mean, even if it were as basic as looking at males, my doubt here is that there's a lot of selection bias in that you're only measuring like the treatment group. But do you have, I mean, and I know it's the field and observation, but would looking at glucocorticoids in females who are not pregnant at the same time, or looking at the profile of glucocorticoids through the year, would that be an adequate control for really making some progress in isolating the extent of glucocorticoid that is attributed to the reproductive event as opposed to anything else?
Yeah, I'm not sure I totally understand which exact result you're talking about, because the microbiome work, or also the increased activity in the HbA axis, this is something we're measuring in offspring. So of course we don't have a control in that sense that we have no reproduction.
No, I was talking about the glucocorticoids that you measure in the females when they are pregnant, when they are.
I do see that. So I think hold on, I have two answers again. So I don't think we need a control. And of course we cannot have we don't need a control, because what we're looking is a continuous variation in prenatal, so in maternal glucocorticoids during gestation. It's a continuous measure, right? And we're relating this continuous measure to later life outcomes. So of course I talked about high and low levels maybe, but of course it's a continuous measure. So, and if offspring of females having lower maternal glucocorticoids levels, having a higher early in development, showing later in life a higher HbA functioning, or having an impoverished gut microbiome diversity and composition. So I don't really see how I would want to have a control for that. You're talking specifically, and maybe this was a bit misleading, because this was just a result we just pulled up for this talk a couple of days ago, with the same time lactating and gestating and only gestating females, and we see differences. This was really only targeted to linking to this 1 year interbirth interval, where we indeed have these two different females, basically. We have females that are not conceiving, that are not lactating, and we have females that are lactating. And there we have this, what you would maybe say, a control group. But in terms of the maternal environment, we don't need a control group. We're measuring variation in adversity, basically proxied as glucocorticoids and later life outcomes.
No, then maybe I missed it. So you've just answered my question. You're saying that you did measure glucocorticoids in both lactating and non-lactating females. My question is, did you measure glucocorticoids in pregnant and non-pregnant females? Because if that's a valid control. If I measure glucocorticoids in non-pregnant females, controlling for season and time, and the glucocorticoids of a non-pregnant female are not different from a pregnant female, my argument that the glucocorticoids are associated with the stress of pregnancy would be invalidated.
So, yes. So we have, so we're doing that. We have very clear, this is again another study though. Yes, we have increased glucocorticoids in different phases. Of course, as Carsten already alluded to, we know that there is variation in glucocorticoids over the reproductive cycle. We have increased glucocorticoids in the gestation. We do know that, and we control for this. We also know, and maybe this is also then answering a bit to Carsten, that we have an effect of an independent effect controlling for female reproductive state of food availability and food intake on female glucocorticoid levels. Is that answering the question? So we can separate.
Maybe I'm getting confused. I still don't see a between subject comparison. The reason I have these questions is because we struggle with the same situation with the owl monkeys. I mean, we will talk in some more at one because it's also seasonal. But because seasonality influences everything we do, if for the same months of the year, I have a female who got pregnant and a female who did not get pregnant, two females, same time of the year, same food availability, same rainfall, same everything, but one got pregnant and the other one didn't. That's what I want to compare. Because if the female who didn't get pregnant now has similar levels of glucocorticoids than the female who did get pregnant, then that kind of complicates the argument.
Let us talk more about that later. I'm not sure
Thank you so much.
I'm not sure. So we definitely didn't do that exact kind of analysis on a monthly basis. So we know that food availability leads to increased conception. We also know that glucocorticoids are predicted by food availability and food intake, basically, in the females. And I have to put that together, what you're asking in more detail.
Yeah. Thank you so much. We have many questions. I don't want to take more time from others. So I think, Tony, you're next.
Hi, I'm Tony DeFury, Department of Anthropology at University of Texas at Austin. Yulia, thank you so much. That was a super interesting talk. And for me, it was very timely. I was just lecturing yesterday about the socioeconomic health gradient in humans and the limited number of studies of wild primates that have kind of looked at that. And so, I've got more data, more information now that I can throw into that lecture. But I was curious because and maybe I missed it. I did have to step out for a second. Did you use rank as did you control for rank in any of your analyses, or what are what's the association of rank and interbirth interval? Is rank predictive of the difference between your shaking head? Rank, talk about rank a little bit.
Perfect. I forgot about it. Of course, I wanted to talk about rank, but rank doesn't do anything. We don't, and this is early when we have replicated it, we don't have a rank effect on food intake. We don't have a rank effect on interbirth interval, nothing.
Not on glucocorticoids and not on kids' glucocorticoids.
No, we do not find rank effects. We do control in some of the papers for it, but still nothing happens. Yeah. This would be it's interesting in its own right, because we have, of course, a situation where there is some food that can be contested for. They are fruit eaters, but all the analysis we run, rank doesn't come out as any, in terms of reproduction, anything important.
And is just a quick follow-up question, is rank very obviously, I mean, is it very obviously linear? Or is it hard to discern relative ranks among females?
It's very obviously linear. It's very hard to get it because these females, there's not a lot of aggression happening. They're following a strategy, I would say, of avoiding each other. They're super spread in the forest. There can be, I think we did it at some point, the average distance between two females is about 50 meters. They are just avoiding each other. They are going into separate food patches. Yes. But they do have ranks. They are macaques after all. They are matrilinearly organized, although the matrilines are much smaller than maybe much smaller than in some other systems we see. Yeah, we have matrilineal rank changes. If there are many females born in a matriline, then this matriline might overthrow the other. We have group splits based on matrilines, so the matrilines stay together. So it does mean something. It just doesn't show up in terms of reproductive rate, basically.
Thank you.
Thank you, Tony. Sorry, I'll skip you for a second. We have a student that I'd like to take first. Leonie Pethig, you're next. If you'd like to share where you're joining us from, your institution.
Yeah, thank you so much, Julia, for this nice talk. I'm a PhD student at the German Primate Center, or basically at the same institute with this very long name as Julia is. So I was thrilled by your result that stressed mothers produce stressed offspring that will develop into stressed juveniles, adults, and then these individuals basically will themselves produce stressed offspring eventually. So this vicious circle you were talking about, did you by any chance run a model that, or to see if you find an effect that your whole population became more stressed? So which meaning had increased fecal glucocorticoid metabolites in the long run, basically. So for instance, I don't know, by just including the date of sample collection over the years or something.
No, we haven't done that. So we're including the. Are we including these? It depends a bit. We're including the year, depending on the exact models we're doing, whether we need it or not. But we have. So I haven't looked into it. We do have a decrease in food availability over the years. This is not analyzed, so this is my hunch more looking at phenology over a long time. I talked to Eduardo about it. This is something we want to do in the next couple of months, hopefully, looking at phenology patterns. So we do see that. And given the correlation of food availability and female food intake and GC levels, there might be something in there. I don't even have a preliminary answer to that, definitely not a definite answer, so I don't know. But so you're coming from transgenerational effects. I think that was your actually motivation to ask the question. So we haven't looked into transgenerational effects because, well, we have to wait until they're actually giving birth and then having grown offspring again. And that doesn't necessarily have to be the case, right? So it goes both ways, of course, because we also have non-stressed mothers producing non-stressed offspring that have a less active HPA axis, basically. But
it could also be
for the-
separated. Yeah,
Yes.
I mean,
Yes.
it could also be a much larger effect of, I don't know, habitat disturbance, anthropogenic impact, climate change, whatsoever. So maybe disentangled from your stressed individuals that give their stress genes basically to the offspring. So yeah, but I was just curious if you looked into it, because with this long-term data, you know, I mean, it's fascinating what you can do with it. So
yeah, so yeah, we haven't done that, and I don't know if so the transgenerational part, I think that is going to be a super tiny data set, so maybe we leave that for others. But you're right. Of course, we climate change is, we only have 20 years of data, but we do see something, and we're going to look into it. And I also was thinking, but I don't know, thinking about which would rapid climate change then hit income species harder, basically, than capital species, because they're relying more on a bit of predictability. And then, so I was thinking more into that direction, leaving the transgenerational bit out at the moment, simply also because it reduces our sample size tremendously. But it's a very good thought, and I will do that. But I was thinking more on the general patterns of what's happening phenology-wise, food availability-wise, and does that influence female breeding decision? And is it, are they maybe buffered more if they have this capital signal, condition-dependent signal? Is that safer, basically, than in an income strategy? So this is, I think, where we're going to think about more in the next time.
okay, thank you so much.
Sari, you're next.
Hello, my name is Sari van Belle. I'm at the university that Tony is at, the University of Texas in Austin, but I'm actually located in Mexico, studying the black howler monkeys, their social system. Thank you very much for your very nice presentation on these macaques. Overall, I have the impression that being stressed is rather has a negative effect. And then there are these mothers that are able to give birth twice a year, causing even more stress. And if I understood correctly, this is only like 20% of mothers that can do that, but please remind me if I'm correct in that. But then comes my question, given that this causes more stress, how is it that this is a strategy? How is it that this strategy can evolve, that mothers would reproduce twice in a year?
So sorry, that was unclear. So they're not reproducing twice in a year. They do either have a 1 year or a 2 year interval, but your question, of course, still holds. Mm-hmm. So they're either they're giving birth April to July, mm-hmm. And if they're in good condition, if food availability is favorable, and if they're had an infant early in that season, and then the next mating season starts in October, but they can conceive until December or so, right? January. So if they have an infant in, say, April, then they might be able to conceive in October at the end of the same year and have another infant next April or so, right? So their interbirth interval is 1 or 2 years. But of course, your question is right. Why would they do that? Again, they only do it in 25% or so. And again, we don't have a rank effect here. It's not the high rankers that are going for 1 year interbirth intervals. We don't see that. Why would they do it? I think the benefit is still very strong in terms of female reproductive output. We see that we have this. So this was the graph I showed on infant survival after a 1 year interbirth interval. It's a bit reduced, but our infant survival is insanely high. So we have a 90% or so of infant survival, and the effect of an interbirth in a short interbirth interval is super small. So in terms of survival, I think it does pay a female to go, if she accumulated the physical condition she can conceive, then it's super beneficial. And then, of course, we started to look, and this is a very quick and dirty first analysis. It seems, and it makes a lot of sense, that there is an added cost to both invest into gestation and lactation, and they have this higher stress levels. And now your question is, why would they do it? I would think, again, trade-off. It's so beneficial to sometimes have an extra offspring that they can live with the higher glucocorticoids. But it's an excellent question. This is why I said it does come at a cost. But this would then open all these questions, like, would you ever want to produce an offspring in a situation where it's not optimal, and then this offspring is in a not as optimal, doesn't grow into this optimal phenotype? So this is, I guess, it's always a decision. But I would say the benefits outweigh these costs.
I have another question. Have you looked at the effect on male infants of all the stress? How does that affect males?
So all the analysis so far are both sexes.
Okay.
So the survival data, and I didn't show growth rate, but the microbiome data, everything is both sexes. And so we haven't looked specifically into sex differences here. So you're thinking, no, I don't know what you're thinking actually. So no, we don't have, so we have tiny sex differences specific. Yeah, sorry.
I'm sorry, I'm getting interested, and I know that you don't have the behavior part, but it would be interesting given that this is the dispersing sex, to see how that affects. That was, okay. And I know that you don't have yet the data, but it would be interesting.
So yeah, it's super interesting. So we know that prenatally stressed offspring, and I said maybe I should, but yeah, there's so much already in the talk. Prenatally stressed offspring, early prenatally stressed, often grow faster. So they have an accelerated growth rate. We do know that. And that might be beneficial actually for males because they're dispersing before they reached full body size. So they are dispersing with around 4 years. This is clearly before they're adults, so they start reproducing with 7 or so. So it might be actually beneficial in that sense. We are predicting, not tested, we're predicting compromised social skills, social cognitive skills, maybe compromised abilities to form bonds. In our system, of course, particularly for males, it's super important to have these strong supportive relationships that help them in rank acquisition, because for males, of course, we do have rank effects in terms of reproductive success. So it might be that, so I would agree there will be, I'm pretty sure we have sex difference there, and we have differential costs and benefits for the sexes in terms of these phenotypic differences that we see. So it might hit a male harder than a female, for example, in terms of being able to form supportive relationships, because the females are nicely positioned in their matrilines, for example, while the males have to go to a new group, have to fight their way into that group, have to form these relationships and so on. But again, we have not looked into that at all.
I came up with a third question. I'm not sure if I should ask it or if I should give the other person.
No, if you can hold it, I just want to be sure. I think we'll be fine because well, go ahead, because I only see Sarah next and we still have 20 minutes. Go ahead, Sari.
Thank you. I was thinking of, and given that I work with howler monkeys, and Eduardo, that is something that you have done with the red howlers, looking at the sexes in terms of the sex of the infant, if there is a change according to condition, population, size, food availability, whether there are more females born versus male borns, if you have looked at that. It would be interesting.
Not at all. So you're thinking Travers-Willard kind of ideas or sex allocation?
Yeah. Females somehow, according to, and I can't remember on the top of my head, but there is a hormone study with black howler monkeys. This is by Pedro Diaz y Ariadna Rangel Negrin, that they have looked at the hormonal conditions of the mother and how likely she will give birth to a male or a female. Anyway, it might be something interesting to explore.
Absolutely, and we're getting at the number of births where we can actually start to look into these effects. Yeah, I think that, yeah, I don't even know. I would have to pen it down first to think about whether I think it's a local resource competition situation or a Travers-Willard more kind of setting. Particularly because we don't have these strong rank effects on the females, so it might not be in that way superimposed. So just top of my head, I would say then females, good condition females, low stress levels maybe should produce sons. I don't know, but thank you. Maybe this is something we're gonna look into, and I don't know the howler study on the GCs there.
Well, you should look into it if you're
yes
interested.
I just noted it.
Thank you.
Thank you, Sari. Sarah, you're next.
Well, hi, I am an undergrad student of the Rosario University, and my question is the next. Well, based on your understanding of the relays income breeding approach in the mixed breeding strategy of macaques, what are your personal thoughts on the potential long-term implications of this strategy on the overall health and survival of the macaques population?
Sorry, I missed part of the question because you were breaking up, at least for me. Can you or someone else?
Can you repeat, Sarah?
What are your personal thoughts on the potential long-term implications of this strategy on the overall health and the survival of the macaques population?
It's also breaking for me. If somebody else can repeat, or if you want to say it in Spanish.
I understood she asked for the long-term consequences on individual health and survival of the.
Is that it, Sarah?
Yeah.
Thank you.
So is this a question? So I'm not entirely sure. So the long-term consequences of population-wide general predictions I don't have anyway. Long-term consequences of prenatal stress. So this would only hit, so this would be absolutely a problem if we are seeing more anthropogenic change, for example, which of course also is a problem in Thailand, even though at the moment it's a very well protected forest. So if we are thinking or decreasing food availability, which might also happen, then we are having increased maybe HPA activity, even further effects on a problematic gut microbiome. with all the downstream consequences. In that sense, of course, we are expecting a lot of health consequences. We are not in the, I don't know how quickly we can pick up on those. Again, we haven't looked into this long-term variation. So anthropogenic change we have not looked into at all, because we always felt so far very lucky that we don't have to consider it. But phenological mismatches might happen, and that might have, of course, consequences in birth rate, infant survival, also, but we are unfortunately not getting into that because we don't have the data to look into this. You could think of if there are more phenological mismatches happening more rapidly now with increasing climate change. We could think about there are more females that are conceiving and not able to maintain it. So in terms of miscarriages, which we don't have the data. Because it would need a lot of hormonal data. We have a lot of glucocorticoid data, but not female reproductive hormones dense enough. But that would be something I can imagine. So increased reproductive failure, maybe increased infant mortality, and there's all kinds of health consequences. We are together with the Primate Center or with the hormone lab at the Primate Center always trying to develop new markers for health that have to be, of course, non-invasive. So the gut microbiome is one. We are looked into other markers, but we haven't analyzed it in a bigger long-term perspective.
Thank you. Amiyal, for those of you who don't remember, our speaker next week. You're next.
Thank you. Thank you for a great talk full of so many interesting details. I got two questions. One is short. It's about a detail that I may have missed. For the microbiome analysis, was it done on only young offspring, or you looked at the effects of GC on older individuals?
Yes, so this should have explained it in more detail, I guess. We're following the exactly same regime as with the HbA study, so with the stress study. So we look at the different age classes. So we're looking at, so it's a cross-sectional study, right? So we are having data on prenatally stressed females. So we basically have data on maternal glucocorticoid levels for not all of our offspring, but particular cohorts of offspring, and for those offspring that we looked in a 1 year snapshot cross-sectionally at their microbiome composition and diversity. And since we are having different cohorts, one from 2011, for example, these would now be, when this study, the snapshot study took place, they are adults now. And we have an intermediate cohort, they are juveniles now. And then we had the new cohort and they were infants. So we have these 3 age classes basically. And this is how and then we did statistical interactions with age, and could then say that this effect on, for example, decreased alpha diversity is maintained into adulthood. So if we see it in the infants, we see the same pattern in juvenile, we see the same pattern in the adults. Does that make more sense now?
Yeah, thank you.
Sorry, I was too quick, I guess.
If I may ask another question, I hope it's not challenging. The last part of your talk reminded me of results on the effects of early life adversity in the Amboseli baboons. I was wondering if you can, maybe make some comparison. Is it maybe a different description of a similar story, or you see these systems as actually different, or you're actually looking at different things?
We're looking at the same things with not as nice data, of course. so we're looking at very similar questions. I would say the big difference is that we are very, very interested in these timing effects. Because this is something we know from humans. We are very interested in the different sensitive phases, and this is why we also consistently find, and I think this is where we have different, at least interpretations maybe, we consistently find an effect of the early, so the first half of our gestation period. Sometimes we don't find an effect at all, like for the microbiome data, at all for the late gestation. And sometimes we find it for the HbA, so for the glucocorticoid analysis, we find a less stable, less strong effect for the second half. And so I think that is the main thing. And the same goes for the I call it the postnatal phase. So you remember we have these three phases: early, late gestation, and post parturition, I guess, early lactation. And we only find our results for the early phase. And the Amboseli project basically thinks of all of that as an early life adversity. So there are many, I'm sure you know, these different early life adversity categories. And then they have, of course, prenatal, but they are not looking into the specific timing. And I think this is something that we need to better understand, because sometimes they might even go into opposite directions, these effects. So you could have, for example, this is something, now I go back to the growth data, which we are, of course, which I didn't present, but this is Andreas Berghandel work. So where we're showing we have an accelerated growth rate with as a function of prenatal stress. And detailed or correctly put, it would be we have if there's early increases in, or there's high early prenatal GCs, we have this accelerated growth. Postnatal exposure. So if the lactating female, basically, if that's high, then we actually have a decrease in growth rate. So these effects can be completely opposite or canceling each other out. And this is, I think, one of the main design, but also conceptual differences that we have in contrast to Amboseli. We're really interested in sensitive phases and the timing effects. Overall, of course, it's the same questions we are asking. So this is absolutely right. We are both interested in early developmental, or in adaptive developmental plasticity, in developmental constraints. So this is all absolutely the same.
Thank you.
I just want, it was my turn, then we'll go to Eyal, but I want to take it because I want to have a follow-up. I'm assuming that, Eyal, you were referring to the paper by Lange et al. Elizabeth presented. I think that most of us look at the baboon project as kind of the gold standard, but what they did is very, very different from what Julia has done. For one, they have absolutely no data on food. Their proxy for food was drought. And they combine 6 factors in one index. One of them is, did they get 200 millimeters of rain or less? So I really think that it's a completely different approach. I agree with Julia. And that's where sometimes I have these conflicts which actually was a question I have for Julia later. How about social factors of those females? But sometimes we feel like, when do we go deep in some possible factors, which is what you guys are doing, Julia? And when do we go broad, produce an index with 6 categories of, in my opinion, doubtful in terms of validity? Rainfall as a measure of food available ought to be checked. And I think that there's value to both, but they're different. I agree, very different. And I like it that they're trying to connect to levels of hormones that we're assuming are related to food. Well, you want to measure food. But just quick question, what is your intake data? Talking about food, food availability and actual ingestion of food. You mentioned you did collect intake data, right? Right, we
Yes.
can do that. Yeah, that's
Yes,
correct.
no, we have. So we have it sometimes. It's not in all analysis. It depends on whether we're looking at, as you just said, it always depends on what you're putting into one study. But we do have, I talked about at least one result from Sonia, yes, that gestating females, for example, have lower food intake. And then in this case, it's really food intake. So we are measuring basically bite rates for each food item several times, and then have an average bite rate. We don't have, that would be gold standard, we don't have individual bite rates for every single item. So we're assuming all adult females have the same handling time, ingestion time. So that's our, I don't know, proxy, I guess. But this is what we're doing. So we have bite rates for the individual items, and then we do nutritional composition. So we know fat and fiber and sugar and so on. And this then linked to feeding time, we can calculate, caloric intake, protein intake, and so on.
Great. So we're going to take one more question, and I'm delighted to see that it's from a student. I thank Sarah, and I thank, I'm looking at the screen if they've left, but all of you, we definitely want to have more students joining us and asking questions. So we're going to take the last question from Eyal Rosengarten. Please, if you would like to introduce yourself, and then we need to give Julia, like I announced, a well-earned break before she goes on to the individual meetings. Eyal.
Thank you very much. Thank you for the fascinating talk. My question is, you mentioned earlier.
Can you let us know where you're joining us from? Where are you?
I'm a master's student in the Bar-Ilan University in Israel. Sorry if you hear noise in the background. It's my nieces and nephews. My question is, you mentioned early on that you had a split of your population, of your the population you are surveying. Did that split, in your opinion, did that split affect the nature of your research in terms of the food availability or any of the parameters you were looking into, as opposed to if the population stayed in a single location?
Yes, so thank you. They're staying, of course, on a larger scale in the exact same location. I showed you this map, right? And I also showed you that the groups are very much overlapping. They're not territorial in that way. They're overlapping in their home ranges. So the group split, the group tend to split. Again, we had now 3 or 4 group split events. So we don't have enough to really look into it. It seems like they're splitting when the number of adult females reach a certain threshold, then they are splitting. So I would argue it has something to do with feeding competition and just expenditure due to travel distance and so on. So that's what they're doing. And they're splitting based on the matrilines, and then the males, they are unsure where to go in the beginning. They swap, and then they finally settle. The females, juvenile females, of course, stay with their matrilines. The males are very often, this is not purely analyzed now, they're going to the opposite group often. So we feel like sometimes they are using it to disperse. And in terms of food availability, so the overall food availability is not affected, of course. They're staying basically in the same home range. They sometimes just distribute the old home range between them, kind of. So nothing really changes. We don't have a high resolution, small scale idea of habitat or of home range quality. So we have a phonology with botanical plots in the entire area, but it's not like we could say, well, this is the better part of the home range compared to that one. And for all the analysis, of course, we are controlling for number of females and so on. So that, of course, would affect the analysis because we're going from, I don't know, 30 females to 15 females or something. So, of course, there's an effect. We also know that more males in the groups does something to home range size. So it has effects. Yeah.
Thank you very much.
Thank you, Eyal. So with that, we're gonna let Julia go and take a well-earned break. But those of you, if I can take 2 minutes from your time, I just want to say something. Julia, thank you so much for your talk. Thanks everyone for joining us. And Julia, you're welcome to come back at 1 p.m. I'll have the link open so that you can meet with you can have your first meeting. Go
Yes.
take a rest.
Thank you very much again for having me and for the discussion.
See you later.
See you.
So, I just wanted to say we have an unusual number of students already at the end of the meeting, and we're the organizers of FINE were talking about this today before. So, Ana Lucia and Nina, I gather your students, and Eyal, Yabonga, we have not heard from you, so I don't know if you're a student or not, and Lindelani, but we're delighted to see this. We've been talking among us that we're still uncertain what we can do to have more students engaging, attending, and participating like you've done. So I just wanted to let you know that if you have ideas or you email me or you want to share something now of what we could do to make FINE more attractive to students, we want to hear from you. We feel like we haven't figured it out. We're not getting as many students as we would like to see, not just attending, but really doing what you guys are doing, which is hanging out and staying for the questions and answers and feeling comfortable being part of the dialogue. So I have time. I'm meeting with Julia at 1, but if anybody has anything that you guys want to share, I'll be very interested in hearing your thoughts.
I'm trying, and I should try more to share this seminar with my students and encourage them to join. I see it as a great community and a great opportunity for students. For example, my university is Bar-Ilan University in Israel. We do not have a lot of relevant visitors from abroad, so this is a great opportunity for my students, for example, to interact with members of this community. I think that if more PIs encourage their students to do so, we will have a larger audience.
You're making me think, would it work, for example, at your institution? I'm thinking myself about the University of Buenos Aires, or Sari was there. Do you know if your institution has an email address that they would like to get the announcement? Because I hear you, Eyal, I have so many things that come in and I want to share with my students, and then I forget, and by the time I remember, it's buried in my inbox. But I wonder if some institutions, some colleagues may say, you know what, send the announcement on the list. We have 800 people on the list. But what if for some people it would work better that the message doesn't go to you, but you tell me, listen, send also a message to this email that circulates announcements in the university?
Yeah, I think it's a good idea. In my case, I'm currently at the general faculty of biology, so I think this message would be irrelevant to almost all labs. But there are many people in this community that are part of, ecology, evolution, and behavior departments, and reaching to these departments and trying to address them, I think can really help. Yeah.
Great. Lindelani, yes, please let me know. I'm just keeping an eye on the chatting. Well, that's all I have. We'll see you guys next week to listen to Eyal present his talk. It'll be exciting to to hear about hierarchies for the first time. So that'll be fun. Thank you so much for joining us. I want to take a little break myself before my meeting at 1. Good to see you, and see you next Tuesday. Thanks for joining us. Bye.